All right, so let's talk about the respiratory system. Respiratory system, what's its function? What's the big kind of goal? Gas, you got it, perfect. It's gas exchange. We've got to get oxygen in, we got to get CO2 out. Our cells produce all this carbon dioxide, and we need to get that out of our blood into the atmosphere. That's the whole kind of goal of this respiratory system. What we'll do is I'll kind of go over the main parts of the respiratory system here. Drawing a little sketch of a person. And I'll get things in a little bit more detail. So here you've just got the profile of a person. Here's a man. All right, his ear, he's got some hair. All right, got his neck, go down to his shoulders. All right. Now, first part of the respiratory system that air enters most of the time is going to be right behind your nose. It's your nasal cavity. So your nasal cavity is obviously right here, and it's this kind of large cavity that air goes into once it goes through your nose. All right. Nasal cavity's got a bunch of different functions, important functions. What do you think it does to the air? It filters it, for sure. We've got nose hairs, okay, and so that's going to filter out some large particles. It also kind of cleans the air using some other techniques. So, first off, on the inside of that nasal cavity, you have these deep folds in the cavity. They're almost like these big grooves. They're called conchae. You spell that with C-O-N-C-H-A-E. And so what happens is you breathe in air and you have these like deep grooves and kind of like irregular surfaces on the inside of your nasal cavity. It causes the air to swirl around, right? Now the sides of your nasal cavity, it's like you have all these epithelial cells that secrete this mucus. And it's this like sticky, wet mucus stuff. So the air swirls around, and that causes all the dust and stuff that's contained in the air to hit up against the side of the nasal cavity, and it gets stuck in that mucus. So it filters it not only by nose hairs kind of filtering out the large particles, but as the air swirls around, some of those particles will get stuck in that mucus. And where do you think that mucus goes after it like collects all those dust, bacteria, viruses, everything else? What do you think happens to that mucus? Yeah, that's right. We swallow it like all day long. In fact, we have a bunch of epithelial cells in the nasal cavity. They're ciliated, so they have little cilia, and they beat in that mucus, and they cause this like current, this lazy river current that just goes back towards the back of the nasal cavity, down the throat, down the esophagus, and then it's perfect because we can just put all that nasty stuff in our stomach acid that kills everything. Okay. Our nasal cavity also is going to warm and moisten the air. So, we have warm body temperatures, and a lot of times the air is not so warm. So as air comes in, it'll get warmed up by the nasal cavity. We have a lot of blood vessels that are close to the surface, close to that epithelium, that function to really warm up that air. It also moistens it. It takes the dry air that we inhale and adds water to it. Okay? All right, we've also got right here, we've got the oral cavity. I'll draw some teeth. Got a little tongue here, right? The oral cavity is obviously right behind our mouth, and what connects the oral cavity to the nasal cavity, so this is nasal, let me label it. This is nasal, this is oral cavity. What connects the nasal cavity to the oral cavity is this muscular tube. It's like this funnel-shaped tube. It makes up our throat. The technical term for it is our pharynx. Okay. So this whole region back here that's behind the nasal cavity and the oral cavity, that's your pharynx. And it's a muscular tube that connects the two kind of cavities. Okay. Nasal cavity, oral cavity, and then the pharynx. The region of the pharynx, it's right behind the nose or the nasal cavity. That's called the nasopharynx. The region of the pharynx is right behind the oral cavity is the oropharynx. And we've got one more part of the pharynx. I'll tell you what its name is here in a second. All right, we're tracing the pathway of air as we inhale. It goes in the nasal cavity, then it goes through the pharynx, then it's going to go through a tube that goes down kind of this cervical region. What's the name of the next tube that the air is going to go through? Before the larynx. Okay, perfect. It goes through the larynx. The larynx is like right here, and what it is the larynx is a series of 9 cartilages. So it's this cartilaginous tube that extends about the length of the neck. It's about 5 centimeters long. It extends from C2 to C5, I believe. But it's about 5 centimeters long. And this larynx has 3 purposes. Anybody know it? Anybody know what it is?
Your voice box.
Voice box, that's number 1. It contains our vocal cords, all right, which are perfect. In fact, the largest of these cartilages, I'll kind of draw it like this, right? It kind of sticks out a little bit further than all the other ones. That's called the thyroid cartilage. Thyroid cartilage. That's your Adam's apple, okay? So that large cartilage that makes up your larynx, right here, that's your Adam's apple. Within that cartilage, we have some like little bands of elastic fibers. So it's connective tissue basically. And if I were to draw a view looking like down the larynx, so it's basically like, you know, a little tube, all right? Imagine this is a cross section of the Adam's apple. If you were to look down there, you would see these like little bands of elastic fibers that kind of go across that opening, that tube. Those are your vocal cords. All right? And those vocal cords, this is kind of how they work. It's pretty cool. They're connected to these little tiny muscles that are also situated within this cartilage. So we'll just say the name of these muscles is not important for this class, but we'll just say they're tiny little muscles that are attached to the vocal cords. And when those muscles contract, it causes the vocal cords to get really tight, and it also kind of changes their position. So when these muscles contract, the vocal cords go from kind of like this orientation all the way to like this orientation. And it blocks off more of that airway. And then we push air through it, it causes those bands to vibrate, okay? And that produces a sound. And we can control how loud the sound is by how kind of forcefully we push air through, and we can control the pitch of the sound by how kind of strongly we will pull on those muscles to make the bands kind of like loose or tight. We can control sound. All right. And then that sound comes up through our larynx and it comes out our mouth, and then we move our mouth and tongue in all different ways, and that produces speech. Okay. Like if you were to put a microphone right up to the larynx, would it sound like somebody talking? Like if you were to just listen to the sound without, no, it would just be tones, you know, because we change all that sound, you know, in our mouth. It's like the instrument. I don't know if like if you're thinking about a trumpet, you know how you like the sound's made at the mouthpiece, right? But then all those little buttons, I don't know how to play a trumpet, but like if you knew like all those little buttons and what you do, that's what produces the music that comes out the end.
So how does that work with like, is it somebody had their larynx removed and they've got the device they speak with? What is?
That's a great, so this is perfect, Justin. This is actually a great way to explain what's going on. So the name of that procedure escapes me. But what happens is they basically have had their larynx removed. They don't have any vocal cords. And so this little machine, all it does is it produces a constant tone, okay? And it just kind of injects that vibration into the throat area. And all they do is they just modulate how that sounds by moving their mouth and tongue. Now, if you notice that it kind of, when you hear someone who uses one of those devices talk, they talk like kind of really monotone and their words kind of like run together. Well, there's two reasons for that. Like number one, your mouth can't really change the pitch of that vibration. That's set by the machine. Now our vocal cords, we can do that just by how tightly we pull on those cords. Another thing that's really important for speech is that we have a really fine control over when we start a sound and when we stop a sound when we're speaking. So we can like turn it on and off really quickly. When you have that artificial device.
Voice box.
Yeah, what's that?
It's called a voice box.
When you have the voice box, you don't have a fine control over the stopping of the speech, right? And that's kind of like the main difference of why it sounds so qualitatively different than an actual talking, right? Okay, anyways, so voice box is in the larynx, series of 9 cartilages, okay? The first cartilage, the one that's way up top, this is a special one too. I'm going to draw him in purple. He is this little like flap of elastic cartilage. In fact, there's only 2 places that our body has elastic cartilage. And you guys remember where they are? Where are the 2 spots that we have elastic cartilage? Ears and epiglottis. Perfect. And that's what this is. So the first cartilage of your larynx is called the epiglottis. It's this guy right here. And what the epiglottis does is that when we swallow, we kind of move the larynx up and the pharynx kind of anteriorly, and that pushes the epiglottis down. It's just like a little trap door that closes off the larynx so food doesn't get in there when we swallow. And then we're not swallowing, it opens back up so air can pass through. Right? Because there is another little tube that's just posterior to the larynx. And it's not associated with the respiratory system. What other tube kind of splits off?
Esophagus.
Yeah, your good old esophagus. And when you're a pharynx, you've got food and air. Now, posterior to the larynx is your esophagus. That's where food goes when you swallow. And the epiglottis is what closes off the larynx. All right, so right here, we'll do the esophagus. Yeah, the epiglottis is just a little trap door. It's made of elastic cartilage that shuts and closes off the opening to the larynx when you swallow. Okay.
So is that like something to do with like acid reflux or something? Like what stops that?
Oh yeah, so you're thinking about those are that's the cardiac sphincter. That's the sphincter that's at the top of your stomach. It separates between the top of your stomach and your esophagus. When that's not fully closed, it allows some of the acid to come back up. The epiglottis is there just to cover up that the airway. Right. All right, perfect. All right, now somebody said it, after we go through the larynx, we got another tube, a cartilaginous tube, goes way down here. That's your trachea. Okay. It's also cartilaginous. It almost looks like you know those things like when a big building is being constructed, it's being built, and you'll have that tube that they throw garbage down, right? It goes down the whole side of the building, and it's like lined with those rings to keep it open. That's kind of what the trachea looks like. It's a tube, but it's reinforced with cartilage that keeps it open. And we'll talk about why that's important. So the trachea comes down here. Once it gets down into the thoracic cavity, it splits into 2 large tubes. Each large tube is called a bronchus. Bronchi for plural. So I'll say bronchus, B-R-O-N-C-H-U-S. The bronchus, they go to the lungs. I'll draw these guys in green. We've got 2 lungs, right? Which lung's bigger, left or right? What's that?
Right has more.
That's right, that's right. So one of them is bigger. In fact, that bigger one actually has an additional lobe. One lung has 3 lobes, one lung has 2 lobes. That one's got 3, right? The right lung. The right lung has 3 lobes. The left lung is a little bit smaller and it only has 2 lobes. Why is the left lung smaller?
Is your heart there?
Yeah, your heart's there. Your heart takes up a rib right in here and kind of crowds that left lung. The lobes of the lung, if you had to guess what their names were, and they're in this orientation, knowing your anatomical terms, what would your guess be? Superior, inferior, and middle lobe. And then superior and inferior, right? So, and that's them. So the lungs is obviously where the gas exchange takes place. That's where we need to get the air. After the trachea, oh, I didn't label the trachea, did I? After the trachea splits into the bronchi, those bronchi then enter the lungs and they start splitting up smaller and smaller and smaller. The bronchi will split up once they get into the lungs. These smaller tubes are called bronchioles, and they just keep getting smaller and smaller and smaller and smaller. So I'll just write it under here. Bronchus and then bronchioles. They're smaller. They're like the smaller divisions. Okay? Oh, the division right where the 2 bronchi split from the trachea, this little spot here has a special name. That's called the carina. And it's loaded with sensory cells, like C-A-R-I-N-A. It's loaded with little sensory cells. Why do you think that's the case? Why would we want a bunch of sensory cells right there?
To ensure you're just getting air or gas in it.
Smelling.
Close. What would we not want to go down there?
Solid.
Yeah.
That's what I'm saying. So just
Yeah.
make sure you're gassing.
Perfect. Yeah, that's good. So it's like basically if anything touches that area, it elicits like the most violent coughing fit ever. In fact, people can be completely non-responsive, but the last thing that they'll respond to is, you know, when their carina is like poked with something. You know, that's like kind of the last part of your body that will respond to a stimulus. And that makes sense because if, you know, something gets lodged down there, you're really in trouble because it's blocking airways. We've also got a lot of sensory receptors all through our larynx. And that I mean, you guys have experienced this 100 times or thousands of times. You're eating dinner, water goes down the wrong way, you start coughing, and you just can't stop for a couple of minutes. That's because that water stimulated all of those sensory receptors in the larynx. The response that would happen if in the carina would be 10 times worse, right? It's kind of like the last line of defense for anything that kind of gets in your lungs. Okay. Good. Now, these little tubes that go into the lungs, they get smaller and smaller and smaller and smaller. If we zoom in to what they look like when they're super, super small, they kind of dead in these. So here's like the last little tube. They'll dead in these little sacs that look like a bundle of grapes. If you were to zoom in on what the lung looks like microscopically, it almost looks like a bundle of grapes. Looks like this, with the smallest little tubes leading to those little grapes. What's the name of those little grapes? Alveoli, right? So this is alveoli. The singular for that is alveolus. A-L-V-E-O-L-U-S. And this is the smallest little dead-end spot in all those little tubes. And this is how gas exchange occurs. It's kind of cool. Running up right next to these little air sacs. All right, so I mean, these things are small. They are about 20 micrometers in diameter, if I remember. Very, very small. And you'll have all these little blood vessels. I'll just draw the blood vessel in black. These capillaries that come in and border right next to the alveoli. Now, what percentage of atmospheric air is oxygen? You guys know? Like if you're gonna think about like the air in this room, what percentage of it is oxygen?
21.
Yeah, you're exactly right, like 21%. All right, so we've got about 21% oxygen. What percentage of the atmospheric air is CO2?
0.06.
Yeah, 0.03. Perfect. So there's not a lot of CO2 in the air at all. Okay, very low amount. So we just took a breath. We have some air from the environment come in, and it finally gets to this alveoli. And we've got tons of oxygen in here. That's these blue dots. A lot of CO2 or just a little bit?
A little bit.
Just a little bit. All right, not much at all. Now, this capillary is carrying blood in this direction. All right. Where's this blood coming from? It came from the heart, definitely the pulmonary circuit of the heart, right? So it came from the pulmonary artery. But where did, I guess, that blood come from? Well, it came from the body, of course. And it's a really bad question. It's a good, bad rhetorical question. The point I'm trying to make is that this is like all the old blood that has a bunch of CO2 in it because it just went, it picked up all that CO2 from all the tissues in the body after they dumped all their waste, right? All these tissues in the body were cranking out, we're using oxygen and dumping off CO2. So this blood that's returning to the lungs has a ton of carbon dioxide in it. That makes sense? Does it have a lot of oxygen or a little bit?
Little.
Just a little bit, because it was all used up. It's just a little bit of oxygen. Now, the lining of that alveoli is made up of a special type of epithelium. Do you guys remember what type of epithelium makes up the alveoli? Simple squamous epithelium. And that's like the epithelium that's super thin, right? Just a single cell layer across. Well, this is why it's super thin. This is perfect. Here we've got a lot of oxygen. Here we've got not a lot of oxygen. Which way is oxygen want to go? By itself without us spending any energy? To the capillary. It's going to diffuse across, right? Like diffusion, it's going to spread out. So oxygen goes this way, and this happens like passively. Happens automatically. Then CO2, which way is CO2 going to want to go? There's a lot in the capillary, not a lot in the alveoli. Yeah. So it's going to go this way. And so then it kind of diffuses out here, and now there's not a lot left. Now the blood that leaves the alveoli, it's got a lot of oxygen or a little bit of oxygen? It's got tons of it. Okay? And that's exactly what we want, And this is a process that happened automatically. Okay? That's good. Now I'm going to introduce to you guys really quick this idea of partial pressure. Partial pressure, and this kind of makes sense. All right, partial pressure is the pressure that a single gas kind of exerts in the atmosphere. All right? So like the atmosphere, we're breathing in all these different gases, and the total pressure is what we measure, like atmospheric pressure. In millimeters of mercury, it would be, what, 760? If we wanted to figure out how much of that pressure is exerted by oxygen alone, it's a really simple calculation. You just take the percentage that oxygen makes up of the atmosphere, which is what? 21%, and you multiply that by the total pressure. So the partial pressure, of oxygen is equal to 760, that's the total pressure, multiplied by 0.21, which is 21%, and that's what, like 160 or something? Okay? And so that's pretty easy. I was just saying, okay, it was 21%, I said 21, multiplied by the total pressure, so the 760 is atmospheric pressure. And that just told us how much of the pressure is exerted by oxygen. The cool thing about partial pressure is that each gas, they only care about the pressure that that particular gas exerts. Like, oxygen doesn't care about what CO2 is doing or what nitrogen is doing. It only cares about what oxygen is doing. And it's going to diffuse from high pressure to low pressure. Okay? So, this is where it kind of makes sense. What's the partial pressure of oxygen in the alveoli? 160, right? Because this is atmospheric air, which is 760 times 0.21. And so the partial pressure of oxygen, and we'll say that PO2, that's how you abbreviate, is 160 millimeters of mercury. Okay? Now, what is the partial pressure? I'm just going to tell it. What do you guys think the partial pressure of oxygen in this capillary that's going to the alveoli is? It's going to be less than 160? Yeah, it's going to be less. In fact, it's 40. It's about 40 millimeters of mercury. So here, the partial pressure of oxygen is 40 millimeters of mercury. It's much less. And we already know that oxygen is going to diffuse from here to there. There's more in here, and this partial pressure just backs that up. What do you think the partial pressure of oxygen is in the capillary as it leaves the alveoli?
It's got to be higher because it's got a higher concentration.
So it's going to be higher than 40, right? For sure.
It's going to be the 160.
What do you think?
Isn't it like 98%? It's like 750.
Is it the difference between the two?
Yeah, well, they're going to meet in the middle. Because diffusion is only going to work if there's a concentration gradient.
60.
What's halfway between 160 and 40? 100. And that's exactly what happens here. So the PO2 is 100 millimeters of mercury. Point there, that's how diffusion works. Diffusion only works when there's a concentration gradient. As soon as enough oxygen diffuses out that they're equal, they stop diffusing. And the point here is that 100, what we could get out of the alveoli, that's a lot less than what we breathed in. You know, it's still good, like we still got a lot of oxygen out, but it's not the 160 that we brought into the lungs. And that's just due to kind of the anatomy of our ventilation system. Fish actually do a much better job. Fish, the partial pressure of oxygen, well, I think it's a little different in water, but anyways, they can get 98% of the water, the oxygen that enters their gills, because they don't have lungs. Like lungs, you breathe air in and then you exhale it out. Fish's gills, it goes in one direction across all of the gas exchange membranes, so they can pull things out at a much more efficient rate, which is beyond the topic of this lecture. But fish do a better job, and birds actually do a better job, because birds have a three-chambered heart. They don't have three-chambered lung. They don't just suck air in and then blow it out. They suck air in, and then on the next breath, the air from one lung goes to the next lung, and to that lung, to the next lung, and then it goes out. And that allows them to pull more and more oxygen out of the air than we can. All right, it's kind of a fun tidbit. But, okay, good. So, that's important. Now, the alveoli are really small. And then one more point I wanted to mention was that, it almost looks like an upside-down tree. Like the trachea is the trunk, and then it just gets smaller and smaller until you reach the end of the limbs. If you were to go outside and look at like an oak tree, how many times do you think the trunk divides until you get to like the smallest little stem at the tip of the leaf? Like how many divisions do you think?
I don't understand the question.
So if you
How many
start with,
branches?
yeah, well, like
4 or 5?
you start with, yeah, no, that's about right. That's perfect. 7, like 5 to 7 was an average. So you start with the big trunk, and that divides once into 2 smaller limbs, and it divides again into smaller limbs, and that happens about 7 times. Okay, from the largest trunk to like the smallest stem. Now for the lungs, if we start at the trachea and then go smaller and smaller, do you think it's more divisions or less? Like more than 7 divisions or less than 7 divisions to get to the alveoli? Less. Turns out it's 23 divisions, right? So that was the point there is that you like
Fibonacci numbers?
what's that?
So are these like Fibonacci numbers?
What do you mean?
Numbers that occur in nature are Fibonacci.
Oh yeah, I don't know. That's a good question. But I don't know. I don't know. It probably could. It could be. I'm sure somebody's written a book or something about it. But the point here is that the alveoli are tiny. You know, that's the only point there. They're super small compared to the large diameter of the trachea. And another thing that's crazy, so if we look at these alveoli, we know that they're wet on the inside. Like alveoli are not dry, okay? And they have to be wet because all those gas need to fuse into the water of the cytoplasm and the extracellular fluid. What happens to small things that are wet typically? Like,
Yeah, bendy.
where they like stick together. Think about like when your hair gets wet, it all like sticks together due to the surface tension of water. That doesn't happen in the lungs, and it's due to a special collection of chemicals on the inside of the alveoli, and these guys are called surfactants. Surfactants are little chemicals that resist the surface tension of water. They coat the inside of our alveoli, and they allow the alveoli to stay open even though they're tiny and wet. Right?
So it's like Rain-X?
Yeah. Yeah. No, exactly right. It's a good way to put it.
Can you say that one more time? What's your factor?
Yeah, so these are chemicals that resist the surface tension of water so that they allow the alveoli to stay open even though they're small and even though they're wet. Okay. This is one of the things that makes it so dangerous when you inhale a lot of water, like during a near drowning or even a drowning experience. It'll wash away some of those surfactants and it'll cause some of the alveoli to collapse. And they obviously, when they're collapsed, they can't exchange gases, and that's a big problem. All right, now we're going to, yeah, go ahead.
That comment that you just made, is that like, does that mean that it's like water repellent or water absorbent? I'm trying to understand that.
So it's dissolved in the water, so it's not like it's a fat that doesn't dissolve. So it's dissolved in the water, but it just prevents water from pulling on itself so much.
Okay.
So that it, because that's why water, like when you get your hair wet, all your hairs stick together because water's polar and the surface tension of water like pulls it together.
Mm-hmm.
Well, it just kind of resists that so the alveoli can stay open.
Okay.
Infants, a lot of times infants that are born premature haven't developed these surfactants yet, and they'll have problems during respiration, right? Because they just don't have the surfactants and the alveoli are closed. We're going to talk about some hardcore physiology right now about how we breathe, all right? I tried 2 times a day to do an online lecture on this very topic, and it just didn't come out right on the online form. So I gave up. I literally tried to record twice, and I just gave up. So I'm going to give it a shot here. It's better in person anyways. So I'm going to talk now about how we get air from here into here. All right? Your lungs and your heart are enclosed in a cavity. What's the name of that cavity? Thoracic cavity. Perfect. All right? So that's our good old thoracic cavity. And to understand how we pull air into our lungs, we have to understand an equation. It's a very simple equation, but it's an important one. And it's called Boyle's Law. All right? And Boyle's Law looks like this. It deals with the pressure and volume of a container that contains a gas or a liquid, just like our thoracic cavity. And it states that the pressure of the gas in that container multiplied by its volume, all right, so the volume of that container is going to be equal to the pressure in that container multiplied by the volume in that container if you change the pressure or volume. So that wasn't a great explanation, but I'm going to give you an example to help you out here. All right, imagine if we have a syringe, like a typical syringe. All right, you got the top of the syringe. All right, kind of looks like this. Here's the opening, and on the inside we have a plunger. Right, with a little, handle or whatever. Like that's your typical syringe, right? This is the cavity. This is the little container that we're talking about. This is a container right here that holds gas, okay? We'll say that, I don't know, this container has a certain volume to it right now, and the pressure inside this container is, I don't know what atmospheric pressure would be, okay? So we'll say it's like 760 millimeters of mercury, because it's open to the external environment. If we were to take the plunger and pull it this way, what does that do to the volume of that container?
Bigger.
It makes it bigger, all right? What does it do to the pressure inside this container? It decreases, okay? And that makes sense. Like when you pull that stopper, you create suction, okay? Air moves in, right? Even if you put it underwater and you pull that stopper, water goes in, right? The reason it goes in is because you made the volume bigger, pressure went down, so it went from 760 to 750 or something, and gas moves from high pressure to low pressure to equalize that pressure, right? This is how it works with Boyle's Law. P1, that's the pressure in the container at time 1, before we move the stopper. V1, that's the volume of the container before we move the stopper. If we multiply these two together, we're going to get a number. That number is going to remain equal no matter what we do, okay? So at P2, or at time 2, what did we do to the volume when we did the stopper? Did we increase it or decrease it? We increased it, all right? So volume went up. In order for this to be equal to that, what does pressure have to do? It has to go down. Okay, perfect. So take home is just that if you've got an increase in pressure, it's going to be a decrease in volume. Otherwise, if you get a decrease in volume, increase in pressure, they could go different directions. Yeah.
So how would you measure the volume of what would in the lungs how do you measure volume in what with the syringe it would be in milliliters, so what would the
Oh, you could measure it in cubic centimeters or milliliters the same way. But this is how it kind of connects, because on the bottom of the thoracic cavity, we've got this big sheet of muscle, right? That's located right there. And it kind of looks like that. What's the name of that muscle? Good old diaphragm. D-I-A-P-H-R-A-G-M. Okay? Now when this guy's relaxed, he's kind of in this curved concave shape with the concave side pointing down. When we inhale, we contract that diaphragm, and that diaphragm goes from being in this shape to basically flat, flatter. So it goes from being like that to being like that. As it flattens, it's connected to the thoracic cavity, so it pulls the bottom of the thoracic cavity down. What does that do to the volume of the thoracic cavity? Increases it. It makes it bigger, okay? If volume gets bigger, what does pressure do? decreases. All right? So now we know that the pressure of the air on the outside of the body is what? 760. Right? Diaphragm contracts, volume gets big, pressure goes down just a little bit. It goes down to, in the thoracic cavity, to like, I don't know, 758, 759. But it does go down. What do we know about air? Does it move from high pressure to low pressure or low pressure to high pressure? High pressure to low pressure. We've got high pressure here, we got low pressure here. That's what drives that air into our lungs during inspiration, okay? We also have some muscles. We've got your external intercostal muscles. These are muscles that connect your ribs, all right? And they're kind of positioned here. At the same time your diaphragm contracts, these guys contract, and they kind of they pull your rib cage anteriorly and superiorly, and they kind of expand your rib cage out from the sides. So you got a diaphragm that expands it out from the bottom, and your external intercostals that expand it out from the sides. And then the air moves in. Now, what causes the air to move out? Yeah. It's typically you just relax those muscles, okay? And then the recoil, the passive recoil of the diaphragm from here to here, and the passive recoil of the external intercostal muscles just passively kind of squeeze the air back out. As it recoils, the volume of the thoracic cavity goes what? Up or down? So as the diaphragm relaxes, it goes from here to here, decreases, pressure increases, so pressure's higher now, it's probably like 761, pushes the air out. Okay? Yeah.
So when people like hyperventilate, what are they doing?
That's a great question. So what you're doing when you hyperventilate is you are offloading more CO2 than you typically would otherwise, right? And your respiratory centers in your brain, they keep tabs on the level of CO2 in your blood, and that's what kind of, modulates respiration. So what you're doing is you're just dropping your CO2 levels in your blood way, way, way down, and the effect that has on, I'll have to get back to you. I forgot off the top of my head exactly how it all works. Okay. Yeah, but you're just basically offloading more CO2 than normal. All right. Believe it or not, this is not the whole picture to the story. All right, this is kind of like the intro to how we breathe. Now, I'm going to tell you guys another kind of version of what's going on. And then we'll go to break here soon. So in your thoracic cavity, it's a little bit more complicated than I originally explained it. In your thoracic cavity, we'll say right here, this is a really simplified version of the thoracic cavity. The thoracic cavity is lined with a sheet of connective tissue. That sheet of connective tissue is called the parietal pleura. Now, if you're going to draw the lungs are right here. I should different color. We'll draw this guy in purple. So the two lungs are right here. The lungs are covered with another type or another layer of connective tissue. This is called the visceral pleura. Visceral Pleura. No, U. No, EU. That's right. All right, good. So again, this is just this is the thoracic cavity, and that green layer, that's the pleura that coats the inside of it, and then the lungs are in black here. The visceral pleura, that's the layer of connective tissue that encases the lungs. Now there's a space between the parietal pleura and the visceral pleura. It's called the pleural space. And that pleural space is tiny. You know, I drew it really big here. So this pleural space is kind of like what's shaded in black. It's the space between those two layers of connective tissue. It's tiny, but I drew it really large. It's filled with fluid. And what's special is that the epithelial cells of the parietal pleura are constantly sucking up that fluid. So they're like constantly absorbing fluid out of the pleural space.
I have a question.
Yeah.
So, isn't pneumonia like fluid on the lungs?
Fluid in the lungs.
Fluid in, is that the fluid that gets in the lungs?
You know, it could, that's a different fluid. It's, I think, so when you get pneumonia, and again, like, I could be a little off here, is that this is when you get a bacterial infection in your lungs, and you'll get a buildup of fluid due to the inflammation, due to the infection. And once you get fluid in your lungs, it just like messes, just like when you drown, it screws up all those surfactants, and you can't respire. So that's what happens there. You just get pneumonia, fluid in the lungs due to an infection, viral or bacterial, and that stops you from being able to breathe in that part, right? This is fluid that's just outside of the lungs, and it's between the lungs and the thoracic cavity, okay? Because water's being sucked out of that pleural space constantly, it creates like a little vacuum, and that holds the lungs right up against the inside of the thoracic cavity. It's almost like two slides that have a drop of water between them. They're like stuck together. Well, that's what happens to the outside of the lungs and the inside of the thoracic cavity. They get like stuck there.
How does it not tear?
What's that? Well, that's a good question, because so it's fluid, and so they can just slide past each other, but they're just stuck there, all right? Now, this is how, and this is really important. So now we take our syringe, but in this case, it's not just open to the outside environment. We've got a little balloon right here. And it's just like this rubber balloon it's looped onto the opening of your syringe, and it's just hanging into the syringe, right? If we were to pull the stopper this way, what's going to happen?
It's gonna pull the bulb down. Make it bigger.
Yeah, is the balloon going to inflate?
Yes.
Yeah, for sure it will. And this is cool because, I mean, we could follow Boyle's law, but basically we just intuitively, you can think of it, your balloon's going to get bigger as we pull that stopper down. It gets bigger because the pressure goes down here, right? It's still high here. Air is going to come in, right, to equalize that pressure. It's going to come in. You're going to inflate the balloon. That's how our lungs inflate, okay? When we breathe, we're increasing the volume of our thoracic cavity, which is the volume of this syringe. Our lungs are like that balloon that's just dangling there. But because they're stuck to the inside of the pleural cavity due to this suction kind of force that was generated by that absorption, our lungs inflate too, okay? It's like that balloon that just inflates. Now, what would happen if all of a sudden we're doing the plunger up and down, that's our diaphragm, there was a hole right here in the syringe? Would the balloon inflate? No, air would just come in right here, right? And our lungs would just be sitting there. That's what happens when you get a punctured lung, okay? So a punctured lung is when you get, or it's like a hole, you can get a puncture in your thoracic cavity, right? It'll open up your pleural space to the outside environment. When you contract your diaphragm, you're increasing the volume of your thoracic cavity, but air just goes in through the wound. It doesn't go in through your mouth, you know? Does that kind of make sense? It's called a pneumothorax. So, P-N-E-U, pneumothorax. What's cool about our lungs, though, is that each pleural cavity is separate. from another. So if you puncture your right lung, your right pleural cavity gets exposed to the outside air, your left lung can still function. Okay? Yeah, so that's the funny thing about, so healing is a problem. It takes a couple of weeks because this suction force in the pleural cavity takes a long time to get reestablished. It just doesn't come back really quick. So after the wound heals and after you've kind of isolated, and separated the pleural cavity from the outside world, it still takes a while for those epithelial cells to reestablish that suction, to hold the lungs to the inside of the thoracic cavity.
So we're showing the cavity as one, but it's actually two individual cavities?
Yeah, they're separated. That's right. They're separated by the mediastinum, okay? So it's one large kind of cavity that's separated in different compartments. And that's perfect because, that way if, I don't know, you get stabbed on your right side, you can still breathe with your left lung. All right.
Her question with hyperventilation is pretty much you're over-oxygenating yourself and you're not allowing yourself to get the CO2 out. You can't
All right,
do much
I
oxygenation,
gotcha.
you can't offload the CO2.
Perfect. Thank you.
I might go to the oxygen part.
Are you guys feeling okay about this? Feel pretty good about how we breathe and all that? Now, forced expiration is kind of funny. When you really want to breathe out as much as you can, like really blow out, what you're doing is you're contracting your abdominal muscles, like your transverse abdominis and your external and internal obliques. And what that does is when you contract your abdominal muscles, you're reducing the volume of your abdominal cavity, which is forcing all your guts upwards, right? So you're forcing your guts to press onto your thoracic cavity, and you're causing the volume of your thoracic cavity to get even smaller because your guts are kind of getting pushed up in there, and that forces air out because volume goes down, pressure goes up, air goes out. Good. You guys want to take a little break? Let's take a little break. All right, so hemoglobin is a protein that lets us carry a lot of oxygen in the blood. It consists of 4 globulin groups, Globulin, and on the middle of those globulin groups, we have a little heme group. So there's 4 heme groups total. Heme is spelled H-E-M-E. And heme contains iron, okay? Contains a molecule of iron. And this whole thing is hemoglobin. And what's cool about hemoglobin is that on each one of these heme groups, it allows one molecule of oxygen to reversibly bind to that hemoglobin molecule as needed. Okay, so one molecule of hemoglobin can carry up to 4 oxygen molecules. Okay, and lo and behold, hemoglobin carries 98% of the oxygen that's in our blood. Okay, so you can just say like hemoglobin, this carries 98% of the oxygen. If we didn't have hemoglobin, our hearts would have to beat 15 times faster or be 15 times larger, right? I mean, so it's really crucial for just our ability to carry oxygen. One thing that's funny about hemoglobin is that its affinity for oxygen changes in different situations. So let me explain that. Affinity. How would you define affinity? What does that mean? Yeah, exactly. It's like attraction for oxygen is affinity, like AFFINITY. It changes based on pH, the amount of the partial pressure of carbon dioxide, and temperature. So we'll say the affinity depends on pH, the partial pressure of carbon dioxide, and temperature.
Is that partial pressure?
Yep, PCO2 is partial pressure of CO2. All right. Hemoglobin, the abbreviation for hemoglobin is HB. All right. So hemoglobin has a high affinity for oxygen when the temperature is down, when the pH is high, and when the partial pressure of carbon dioxide is high. No, low, excuse me. When the partial pressure of carbon dioxide is low. And so this is what happens. This is kind of tricky stuff, but this makes a lot of sense. So when we take in, what's the best way to describe, best way to explain this? We'll explain it like this, all right? When temperature gets really high, like after you exercise, when you exercise, you're releasing a lot of heat at your muscles, okay? And that causes temperature to increase. What is that going to do to hemoglobin's affinity for oxygen when temperature goes up? It's going to decrease. So it has a low affinity for oxygen. Let's say that a hemoglobin molecule was carrying 4 oxygen molecules. It goes through a part of the body that's really hot, like our leg muscles when we're on a jog. Is it gonna, the affinity for oxygen is gonna go down? It's gonna let go of those oxygen molecules, okay?
So should we think of it, it's like free oxygen or available oxygen? The hemoglobin or the oxygen that's found in the hemoglobin?
Yes, yeah. So hemoglobin's carrying like 98% of the oxygen in our blood anyways. It's all there, 98% is stuck to hemoglobin, as it circulates through our bloodstream. So when the hemoglobin that's carrying all this oxygen, when it gets to a part of the body that is experiencing intense exercise, like our leg muscles when we're on a jog, temperature is way up. That causes the affinity that hemoglobin has for its oxygen. It's not holding on to them as tightly, and so it kind of lets them go, right? That's perfect, because as soon as hemoglobin lets go of those oxygen molecules, they can diffuse across the epithelium of the capillaries, diffuse into muscle cells, and give the muscles the oxygen that they need, especially now that they're exercising really hard, What do you think the concentration of CO2 is going to be like at the location of those exercising muscle cells? It's going to be like high or low? Like super high, because they're burning up all that ATP, or they're, they're consuming all that ATP, making ATP, they're respiring, producing a lot of CO2. If there's a lot of CO2, what's that going to do to the affinity that hemoglobin has for oxygen? It goes down even more, so it lets off more. That's perfect, because the leg needs it, or the leg muscles need it. Then if we have a lot of CO2, do you guys remember the buffer system that exists between carbon dioxide, water, carbonic acid, and the carbonate ions? Do you guys remember that? It was this buffer system right here where if you mix CO2 and water, those guys go into equilibrium and it produces carbonic acid, which is then equilibrium with hydrogen ions and the carbonate ion. Do you guys remember that? It's like this. Well, anyways, that was like the first day. So whenever you have carbon dioxide and you mix it with water, which is the case in our blood, okay, it's going to go into this equilibrium. If you increase the amount of CO2 in a solution, it's going to kind of cause this whole equilibrium to get established. and we're going to get more and more hydrogen ions in the solution because everything's in equilibrium. Does that kind of make sense? Right? I mean, the take-home message here is that if you dump CO2 in an aqueous solution with water, you're going to get more hydrogen ions due to this equilibrium that naturally sets up. What's that going to do to the pH? Go down, right? More hydrogen, you get lower pH. So if we get a lower pH, that's going to cause the affinity to go down even more, okay? So the key here is that when hemoglobin gets to the tissues, you're going to have more CO2 at the tissues, you're going to have a lower pH, temperature is probably going to be a little bit higher, it's going to offload all that oxygen. Then, as it goes back to the lungs, the CO2 kind of goes down, temperature goes down, and the pH is going to go up. Hemoglobin's affinity for oxygen increases, and that allows it to grab oxygen out of the alveoli. Kind of. We'll grab it out of the blood that gets absorbed. So that's like a good thing, okay? All right, perfect. Now, a funny thing that happens is that basically this whole, what I just talked about, is just a nice way that allows the body to pick up oxygen at the lungs and deliver it to the tissues. Okay, it's just a little trick for that. Now, most animals, humans are the only exception. When animals go to live at a higher altitude, their hemoglobin changes in a very specific way. So, where is there more oxygen? Like here at sea level or at the top of a tall mountain? Yeah, for sure. Like pressure's lower on the top of a mountain, okay? The atmosphere is still 20% oxygen at the top of the mountain. There's just less oxygen up there, right? It's just a lower pressure. So there's lower oxygen up on the top of the mountain. When other animals go to live on the top of a mountain, their affinity, their hemoglobin molecules develop a higher affinity for oxygen across the board. Why do you think that is? Or why would that help them out?
Because when they come back, or like, same thing like athletes, right? Like whenever they go and train in like higher altitude areas, like their blood takes it in better.
No, that's good. This relates to that. This is good. Just think about before they come back, right? So you're exactly right. It does take it in better. So if you have a higher affinity for oxygen, that means you're going to be better at sucking oxygen out of the air, okay? You go to a place where there's less oxygen, you're going to want to be able to suck as much out of the air as you can, okay? That's what most animals do, and it works out well for them. Humans are the only exception. When we go to live at high altitude, this doesn't happen like overnight, it happens over the course of like weeks and months. Our hemoglobin actually develops a lower affinity for oxygen. We're like the only species that does that. It's unclear why humans do this, but the thought is that it's the body's way of trying to deliver all the oxygen that we can pick up to the tissues. So if its affinity for oxygen goes really low, hemoglobin is great at like giving it up. So the thought is that like whatever oxygen we can pick up, we just dump it right on the tissues, okay, when we're training at altitude. Now, so you mentioned a great point. When athletes will go train up in Colorado, right, for a couple of months, and they'll come back. So they go to Colorado, their hemoglobin develops a really low affinity for oxygen. Then they come back down to sea level, for the event. There's tons of oxygen at sea level, so now, even though their hemoglobin has a really low affinity for oxygen, it doesn't matter because there's so much oxygen in the atmosphere, they can just pick it up anyways. But the affinity stays low, so they're dumping an even more amount of oxygen at the tissues. Does that kind of make sense? It's like you don't really need a high affinity when you're here at sea level. It's okay if you have a low affinity for oxygen because there's so much, and so your blood just gets super saturated with oxygen anyways, and then whatever oxygen's in the blood just gets dumped all over the tissues. You're delivering more blood or more oxygen to the tissues than you normally would.
And that makes you better performance?
And that it facilitates, it's better performance because you're dumping more oxygen on your muscles than you would normally, right? By tweaking your hemoglobin, that little trick. You also develop more red blood cells. It's another kind of adaptation, or no, another way that we acclimate to high altitude is that we produce more red blood cells. And so you come back down to altitude, you have tons of red blood cells, and those red blood cells are really great at just dumping a bunch of oxygen on the tissues.
So is that why the Olympic Village is in Colorado?
Yes, it's exactly right. Yeah, exactly right. So they go up there because they want those changes to happen to their hemoglobin and their red blood cells. It's pretty cool. All right, we're done with respiration. All right, so take a breath. So this is how we're going to do reproduction. The female reproductive system, what I've done is I've posted 2 group activities online on Canvas. I want you guys to read through those and do those on your own. All right, I'm not going to lecture on the female reproductive system. The things that are going to be on the exam for the female reproductive system are the different hormones that control reproduction and how they change during the menstrual cycle. Say that one more time. The different hormones associated with the female reproductive system and how they change during the menstrual cycle. That first group activity is all about that, and it's really good actually. It's a great way to learn that. It doesn't take too long, it takes about a half an hour, but just between now and the final exam, make sure you read through that.
So it's a group?
It's a group activity, but you can do it independently.
Okay.
All right, you don't have to turn anything in. It's just a nice way to kind of go over that information. All right.
Reproductive, final exam, PowerPoint.
And so I know it's all thanks to Irma, right?
Tuesday night.
So there's another activity. So I'm posting 2. One's on the menstrual cycle and one's on very early development. Just be familiar with where the major events of fertilization occur, like where fertilization occurs, where eggs are released, where early development occurs, and that's all in that worksheet activity as well. I know you guys are kind of really overloaded here on the back end, so I'm going to lecture real briefly here, not too long, about the male reproductive system. Give me like a half hour, and then we're going to call it. All right. Let me erase this stuff here.
Are you good for stuff?
How many questions are on the final?
70 or 80, I can't remember. That's right, multiple choice. What's that?
Yeah, no study guide.
All your study guides, you know, all your notes, all those good notes you took.
You said we could bring our notes?
What's that?
You said we could bring our notes?
No. No, I said your study guide is just, I would refer to your notes and just really focus on the major topics that we talked about in class. All right.
Will our quizzes that we took help any of these questions?
You know, the quizzes, like the quiz questions aren't going to be on there, but if you missed a couple quiz questions, you know, just be familiar with why you missed them type thing. All right, because that could help. All right. So with the male reproductive system, you guys have already seen a lot of this during, you know, Bio 1 and 2 and all that. We're not going to go over the names of all the different structures, but we are going to focus on the process of how sperm are made, because that does kind of relate to, I don't know, some of the important concepts in physiology. All right. So, is the male testicle. Okay, this is where sperm are made. It is a organ that's outside of the body. Why the heck is it outside of the body? Temperature. Okay, the ideal kind of temperature for spermatogenesis, which is the creation of sperm, is 3 degrees lower than body temperature. So I think it's around 96, 97. All right. Sperm can still be made at 98.6. It's just not as efficient. So evolution really selected for the site of sperm to be made to be located outside of the body, even though it has a complete lack of protection from injury. All right. Which is kind of crazy because there is that trade-off, right? I mean, here you have, an organ of the male body that's so crucial for keeping, the human race, the human species kind of going. But selection has pushed for an external location. All right. So within the testicle, you have kind of a smaller oval-like structure. This is actually the testes. And the testes are composed of a series of little tubes. I'll draw these guys in purple, and they almost look like little loops. All right. And these little loops are called the seminiferous tubules. All right. Seminiferous, S-E-N-I-N-I-F-E-R-O-U-S, tubules. And this is where the sperm are actually produced. All right. I'm just going to kind of give you an overview of the gross anatomy of what's going on here. And then we'll talk about how it all happens. Then all these little tubes, all these little seminiferous tubules, they come together into this kind of complicated network of tubes right in here. The name of that structure, I'll kind of draw it a little bit thicker so we can tell the difference here. I'll add some blue there. There. This is called the rete testes. Then, grab a different color. Then this tube, and this was all inside this larger oval called the testes. Then a tube comes out of this guy. Kind of loops around, comes down here, gets a little wider, and stops right about there. This tube is called the epididymis. E-P-I, D-I, D-Y, M-U-S. Then, when the epididymis ends, it forms into a thicker tube that's more muscular that comes out of the testicle, goes to the rest of the body, and you guys know what that's called. That's your, what's the last tube? Vas deferens? All right, V-A-S-D-E-F-T-R-E-N-S. All right. Okay. The epididymis has a couple of different parts. This part up here, this is called the head of the epididymis. This part right here, that's the body of the epididymis. And then down here, this is the tail. Whoa. So, this is what happens. Sperm are produced where? In the testes. In what tube? In the seminiferous tubules, okay? They're produced there, but they're super young, all right? Just super young, can hardly swim, And so there's this like really slow current of fluid in these tubes that flows from the seminiferous tubules all through the root testes, then it flows up into the head of the epididymis, down in the body, and then it finally ends here at the tail of the epididymis. Sperm will start in that little current, and they will just kind of float along, and while they float along, they kind of like learn how to swim, they develop, they mature, okay? And by the time they get to the tail of the epididymis, they're pretty much mature and they're ready to do their thing, right? It takes them 3 weeks to travel from the seminiferous tubule to the tail of the epididymis. It's like a 3-week journey, and in those 3 weeks, they're maturing. Once they get here, they're mature sperm that are ready to, impregnate an egg. They're ready to fertilize an egg, all right? This is where, in the tail of the epididymis, this is where mature sperm are stored before they're needed. When it's time for them to leave the body, okay, you'll get smooth muscle contractions. So in this tube, it's much thicker, and it's made out of smooth muscle. You'll get peristaltic contractions, which are these like rhythmic waves of contractions that will propel the sperm out of the testicle and outside of the body, okay? So the sperm don't swim out of the body, they get propelled through rhythmic contractions in the vas deferens. Yes?
How long can they stay stored there?
That's a great question. What do you guys think? So how long can sperm stay in the tail of the epididymis?
3 weeks. 3 more weeks.
3 months.
Oh.
Right? Yeah, a long time. So they can hang out for 3 months and still be healthy. After 3 months, what do you think happens? They do die,
They get flushed out.
and they get reabsorbed, right? So they just get reabsorbed, right? Everybody just kind of reabsorbs them, recycles those proteins and stuff. A vasectomy is, question?
Oh, I was going to ask about a vasectomy.
Yeah, that's cool.
Where do they go?
Right here, vas deferens. And so they cut it. They'll go in there and they'll cut it and then burn it and cauterize it, and so that it just severs, the tube. There's no exit plan.
They just get reabsorbed every 3 months.
That's right. They're just constantly reabsorbed. So that's right. Sperm are still being produced. They just stuck right. They're just trapped right there.
But
Right.
can't they still get past it? Isn't that how people, even though they get that removed, they can still produce?
Yeah. So what happens there is that it's rare, but it's possible for the vas deferens to kind of reconnect. All right, over time. It's very rare. Typically, this happens within a couple of days or weeks after the surgery, where if the two ends of the vas deferens are close enough in proximity, they'll go back together, which is crazy. And that's why after a vasectomy, they have to, make sure that that hasn't happened. But, yeah.
So when you ejaculate, nothing comes out?
Oh, so the semen comes out, but no sperm, right? And so we haven't really talked about all the accessory organs of the male system. So you've got a bunch of organs that are all located outside of the testicle. As those sperm are removed from the testicle during ejaculation, they get mixed with all types of other fluids, fluid from the prostate, fluid from the seminal vesicle, fluid from the bulbous gland. I forgot them off the top of my head. I should know. But anyways, that fluid has a bunch of different functions. It the first fluid that is released right before ejaculation, it actually neutralizes. It neutralizes the pH of the urethra and sterilizes the urethra. The semen actually is pretty crazy stuff. It contains a lot of sugar. Why do you think it contains a lot of sugar? Energy for the swimming sperm.
It needs a sterile channel to flow through, like semen needs to be clean.
Yeah, well, so the thing is, what'll happen is that it'll, the urethra, what else goes through the urethra? Urine. So that's pretty acidic stuff. So you'll have a basic kind of solution that neutralizes the pH, right? And that's good because you don't want the sperm to be harmed by that. It'll also sterilize the urethra from any bacteria that could be there, and that could help the sperm out as well. So this is kind of crazy. It has some immunosuppressants in it. Semen has immunosuppressants in it. Why the heck would we want immunosuppressants in semen?
So it's not attacked as a foreign substance and killed off before it reaches the egg.
Yes, perfect. Because otherwise, yeah. So Justin was saying that your body's going to attack any living cell that's not your own. Okay, so the female's body, the immune system is going to see these sperm as foreign invaders and attack them and kill them. That would be bad for procreation. So there's immunosuppressants in the semen that deactivate the female's immune system so that the sperm aren't attacked. It also contains hormones that cause the cervix of the female to relax, and it causes the vagina to undergo peristaltic contractions that help propel the sperm into the uterus, right? So there's actually chemicals in the male semen that cause these pretty dramatic changes in the female's physiology right off the bat. There's these coagulation factors in the semen, and this is actually really crazy, where right after ejaculation, it causes the semen to get really thick for a couple of minutes, and then just after a couple of minutes, it gets really fluid and watery. And the idea there is that some of the sperm are a little late to start swimming after they leave the male body. And so the idea there, this is crazy, this is all like evolution and physiology. So what the semen does is it gets like really thick, and it holds the sperm in the vagina so they don't just fall out, and it gives them a couple extra minutes to get swimming, and then it gets all watery so now they can swim up. It's crazy. It's like, yeah, too much. All right, and it also contains, yeah, that's it. That's it. I think we named them all. All right, so back to this. Vasectomy cuts the vas deferens. They hang out in the tail of the epididymis. They can hang out for 3 months. It's like a 3-week journey to get from the seminiferous tubules to the tail. We've got some endocrine glands in the male testicle. We've got Leydig's glands, or Leydig cells. I'll draw these guys in pink. These are clusters of cells. Leydig, Leydig, Leydig's glands. These are clusters of cells that secrete testosterone. Okay? So that's where testosterone production comes from, or these Leydig's glands. Testosterone, that's right. All right. There's a very large artery that feeds your gonadal arteries that will come into the testicle and bring all the necessary blood into this area. It's a kind of an interesting arrangement. So if we want the temperature here to be cooler than the temperature of the body, it doesn't really make sense to pump a bunch of warm 98.6 degree blood down there straight away, right? It would just warm this area up really quick. So there's this interesting arrangement of the artery and veins. So the large artery comes down like this, and the veins that carry blood from the testicle to back to the body are these tiny little veins that wrap all around the artery on their way up. And this is pretty cool. So what happens is that the blood in these veins is cold, okay? It's around 96 degrees. As it wraps around this warm artery, what's going to happen to the blood in the veins? It warms up, okay? By the time this blood gets back to the body, it's been warmed up to 98.6, okay? Now, that's perfect because we've warmed up the blood before it goes back to the body. Now, the warm blood that's leaving the body, it's 98.6 here, but now it's being wrapped around by all this cold blood in the veins. And it's going to do what? what's going to cool down on its way down, okay? This is called a countercurrent exchange system. Countercurrent exchange. And what that does is it allows two parts of the body to be at different temperatures and maintain that difference in temperature automatically. Countercurrent, so like counter and then current, current, and then exchange. Okay. It allows two regions of the body to be at different temperatures, and for that difference in temperature to be maintained automatically.
So the veins are cooling the artery and the
Arteries.
is warming the vein.
That's right. The same thing happens with seagulls, not around here, but in cold climates, you'll see a seagull, like a winter day, standing in icy cold seawater, and its feet are freezing cold, but it doesn't matter because it has this same system where the blood that goes down to the feet gets cooled down, and then as the cold blood goes back up to the body, it gets warmed up before it gets to the body. So you're never really getting like cold blood cooling down the body. If that makes sense. Anyways, take home message, that maintains the difference in temperature. All right? We need that. All right, we're almost done here. I'm going to zoom in on one of these tubes, and we're going to talk about exactly what happens in the seminiferous tubule. All right, so here's the inside of a seminiferous tubule. On the outside of this guy, you have a thin layer of muscle cells called myeloid cells. You label this as a myeloid cell, M-Y-O-I-D. Why the heck would we want muscle cells on the outside of the seminiferous tubule?
Transport.
Yeah, perfect. Yeah, guys, that's exactly right. They just kind of squeeze, and they cause that lazy river to kind of flow through the seminiferous tubule. All right, the sides of the seminiferous tubule. On the outside, you have these special cells. We'll draw one right here. This guy is called a spermatogenic cell. Do you guys remember good old mitosis? Remember mitosis is when you have a cell divide, you get two equal parts. What's the process in which we produce gametes through cell division? Good old meiosis. All right, I'm not going to go a big review into meiosis. M-E-I-O-S-I-S. Meiosis is when you take one cell, okay, and how many cells are produced? 4. Okay? How many copies of the genetic code does each of those 4 cells have?
23.
One. It has one copy. One copy each chromosome. That's right. It has 23 chromosomes, all right? That's a fundamental difference. When you undergo mitosis, all the daughter cells have 2 copies. Like they're just like the parent cells. But when meiosis occurs, you get one cell that undergoes a series of cell divisions to produce 4 cells, and each of those 4 cells are haploid. Okay, so you get one cell here, which has 2 copies of the chromosomes, 2N. It undergoes 2 series of cell divisions, and it produces 4 daughter cells. And each one of these are haploid. They only have 1 copy, 1N. Why do they only have 1 copy? Who are they going to meet up with? An egg. Another haploid who also has 1 copy. 1 plus 1 equals 2. That's the baby. That's the zygote. Okay, so that's the important part, right? You get 1 copy, right? Because you're going to meet up with the other haploid gamete from the other from the other person. All right, sorry, a little long. So this is what happens. Meiosis occurs right here in the wall of the seminiferous tubule. You'll get one spermatogenic cell. It undergoes the first cell division, that's meiosis 1, to produce 2 cells right here. Then we undergo meiosis 2 to produce 4 cells. And guess what? These little cells, those 4 little cells that are produced by that round of meiosis, they have these little tails to them. And that's your sperm. Okay?
I thought the mother.
What's that?
My mind just got blown. I thought the mother did that.
Well, the mom does her own version, right? So
Okay.
she's producing eggs,
Right.
right? And so she has this collection of eggs in her ovaries, basically, that are then released into the fallopian tube, right? And that's where fertilization occurs. So these sperm, like the male, have to do their part. They have to produce their gametes, and the female produces her gametes, and they unite right there in the female's body. One more part to this, and that's that the walls of this seminiferous tubule also consists of these cells that kind of are positioned around these dividing spermatogenic cells. And the only reason they're there is just to kind of make sure that they're dividing in the right direction. They're like placeholders. These guys in green are called sustenocytes. Sustenocytes. S-U-S-T-N-O-C-Y-T-S. And their purpose is just to be there. They're just to make sure that they're dividing in the right direction. And this is happening on both sides. We've got spermatogenic cells that are turning into sperm. And they're flanked by a bunch of sustenocytes.
And then just keep them in place and make sure.
Yep. They just make sure that when this guy divides, he's dividing towards the middle. You know? So then this sperm, once he's kind of produced, he's a young sperm, he goes to the middle. I'll kind of draw this out really quick. You've got this middle pathway down the middle of the seminiferous tubule. That's called the lumen, L-U-M-E-N. And then the sperm floats down the lumen on its 3-week journey to the tail of the epididymis. Sperm are pretty, this is the last thing I'm going to talk about. Sperm are pretty simple in their structure. They consist of a head. They have a body right here. And then they have a tail that's made out of a flagella. On the head of the sperm, they've got a little helmet. Protective helmet, looks like that. That guy's called the acrosome. This is the head. This is the body. The body is packed full of mitochondria. Mitochondria. Why do you think it needs a bunch of mitochondria? What's that? Energy, yeah. It needs to crank out a bunch of energy and power this flagella right here. That's what whips around and lets it swim forward. The acrosome, this helmet, contains a bunch of lysosomes. Do you guys remember what lysosomes were? They're an organelle that does something specific. They digest and eat. Okay? Why the heck would you want that in the helmet? In the
Doesn't that paralyze the female attack system or something?
No, you're close. You're close. It allows it to eat through the egg, the wall of the egg, right? It allows it to dissolve the cell membrane of the egg so that the sperm can deliver a very special package. What is like the main package that it has? It has DNA, right? In the head, in the nucleus. So it has a bunch of DNA right here. And that's important, because that's the genetic code that will be used to create the zygote. All right, we're done. Okay. Thanks for bearing with me. The last lecture was rough, guys. But Irma's fault. It's not my fault. It's Irma's fault.
We blame Irma. Heavily.
Yeah. Yeah, you good.